Okay, so here's our finishing up the review for exam 4. Again, I'm sorry that the live stream didn't work. I didn't realize that I had to have software for it. So I'm just going to be doing this video and get to talk to myself. That's fun. Also, I was going to try and write on a whiteboard, but apparently it flips the words, and I don't think any of you can read backwards. So I'm going to go slowly. I'm going to try and take my time and just go through the rest of chapter 28 and 29. That's what we didn't get to. And let you know kind of what I think are the most important parts from that so that you can study. So starting with chapter 28, the female reproductive system, you should definitely know what an ovarian follicle is. An ovarian follicle, if you remember, is both the oocyte and the follicular cell, the oocyte being the female gamete, the egg cell. You should know the 7 stages of ovarian follicles, starting with the primordial follicles, which is just a primary oocyte and follicular cells. There is the primary follicle, which is a primary oocyte and granulosa cells. And again, this is the time to remember there are primary and secondary follicles, and there are primary and secondary oocytes. So make sure you're clear about which ones we're talking about, or you're talking about. So primary follicle with a primary oocyte and granulosa cells, that's going to be starting to secrete estrogen to prepare the uterine lining for implantation. The secondary follicles still have a primary oocyte, but then those granulosa cells develop into theca cells, which begin to secrete androgens, your estrogen, your progesterone. Then you have an antral follicle, which still has the primary oocyte and more antral fluid. Remember, the antral fluid is what will start to swell and push the oocyte to the side of the follicle for ovulation. After the antral follicle, it's the mature follicle. That then finally has a secondary oocyte, which is developed. After ovulation, the mature follicle, once it loses the oocyte, becomes a corpus luteum, which will start to secrete estrogen, progesterone, and inhibin, which are all hormones to help regulate the cycles to prepare the uterine lining. And if you remember, inhibin is going to inhibit GnRH and FSH to limit follicular development. So that way you don't have too many eggs being released at once. The corpus luteum will eventually regress into the corpus albicans. That's the 7th follicle, and that is mainly connective tissue. It's just going to be regressed and then absorbed back into the cell, back into the ovary. So for oogenesis, that's the actual development of the oocyte. So you have the primary oocyte going to a secondary oocyte, talking about the eggs. Before birth even happens, you have the oogonia, the primordial germ cells in the fetus. Those are going to be going through all of their mitotic divisions, dividing and dividing, until you have a bunch of primary oocytes, all about like 1.5 million of them, all just waiting. In childhood, so that's all before birth. Before birth, you have the primordial germ cells developing into your primary oocytes. During childhood, there is what's called atresia, which is a regression of some of those primordial follicles. So instead of having the 1.5 million that are present at birth, by the time a female reaches adolescence or puberty, that number is much decreased. So after childhood, after atresia, then you go into puberty and menopause, which we've said is regulated by GnRH, simulating FSH and LH to initiate follicle development and egg development. So then you can go into the ovarian cycle. So there are 3 phases. There's the follicular phase, the ovulation phase, and the luteal phase. So the follicular phase, remember, every month it starts with 20 primordial follicles. Only one of those will actually develop into a fully mature follicle that gets ovulated, unless 2 happen to be ovulated at the same time. That can happen. Oh, my connection lost. Let's see if it still keeps recording. So after, I'm just going to assume, okay, yeah, I think it's still recording. After the follicular phase, you then have the ovulation phase, which is stimulated by a peak in LH and luteinizing hormone. After ovulation phase is the luteal phase. That's when the corpus luteum begins secreting estrogen and progesterone to help support the uterine layer. This is also potential time, if there is no fertilization, when the menstrual layer will be shed and the sloughing off of the menstrual layer. That then goes into the uterine and the menstrual cycle, the uterine cycle, menstrual cycle, whichever one you want to call it. Again, 3 phases: the menstrual phase, the proliferative phase, and the secretory phase. The menstrual phase is that sloughing of the functional layer, the sloughing of the functional layer of the uterus, of the endometrium of the uterus. Remember, the endometrium has both a basal layer and a functional layer. The functional layer is the one that is replaced every month. The proliferative phase is the development of the new functional layer when it starts to increase and those glands and vessels begin to grow. And then finally, you have the secretory phase. The proliferative phase, which is just the development of the new functional layer, and the secretory phase, which is an increase in the vascularization and the glands in the functional layer. Again, still preparing it for implantation, ready for a new egg. After that, you can get into some of the accessory reproductive organs, the uterine tubes, the uterus, the vagina. Definitely know what all of them are useful for, where fertilization occurs in the ampulla of the uterine tube or the fallopian tubes, whatever you want to call those. Within the uterus, you need to understand it's for implantation, support, and protection for the developing fetus. It has its 3 layers. Again, that endometrial layer, that most innermost layer, is going to have 2, both a basal and a functional layer. The myometrium is what's going to actually cause the muscle contractions. And the vagina. It's a thick-walled tube used to go from the uterus to the outside of the body. And it has acidic secretions to help prevent pathogen entry, which also then in turn inhibits sperm entry. And your mammary glands, understanding they secrete breast milk and colostrum. You should know the difference between colostrum and breast milk. Colostrum being what's first produced, really high in IgA, laxatives, but low in fat, whereas milk tends to be higher in fats and proteins and other antibodies. And that, I think, is pretty much it for the female system. So we can go into male system then. It starts with the testicles, the 3 cells within the testicles themselves. The sustentacular cells, which secrete ABP, androgen binding protein. The androgen that they're going to be binding is testosterone. Testosterone is released from interstitial cells in the testicles. And then you have spermatogonia, which actually develop into sperm. So 3 cells in the testicles, the sustentacular cells, which are similar to the follicular cells in ovaries. They're going to be supporting, providing nourishment for the developing sperm cells, as well as secreting ABP. Then the interstitial cells, which secrete testosterone, and then your spermatogonia, which develop into sperm. There are 2 processes that happen with each other, androgenesis and spermatogenesis. So androgenesis being the production of testosterone and the increasing the levels, the concentration levels of testosterone in the testicles. So as the interstitial cells secrete their testosterone, sustentacular cells are secreting androgen binding protein, and that protein is going to bind onto the testosterone so that it stays in the testicles. Instead of being absorbed into the bloodstream and moved all throughout the body, it's going to keep it in the testicles. And as those levels of testosterone increase, that will then stimulate the spermatogonia cells to develop into fully sperm cells. They will also, there is that negative feedback though. The sustentacular cells will also secrete some inhibin to help inhibit the development of more sperm cells. The spermatogonia, so that process of developing from a spermatogonia into a fully formed sperm, it's self-divided into 2 processes. So first you have androgenesis, which increases testosterone levels so that spermatogenesis can happen. And then spermatogenesis goes until spermiogenesis, which is kind of the refining of the sperm. So when you go through spermatogenesis, you have a mitotic division so that you can keep the spermatogonia cells the same, because remember the testicles are producing 100 million sperm cells a day. So you need to have the spermatogonia cell numbers staying the same. You have 1 primary spermatocyte that gets developed, and then it goes through a series of meiotic divisions until you eventually have 4 spermatids, which develop into fully formed sperm. And then you have the process of spermiogenesis occurring. So all of this spermatogenesis that happens in the seminiferous tubules in the testicles, and then once they become sperm, once they've gone through the spermiogenesis, and or I'm sorry, once they become sperm, they get moved into the epididymis, which is the tubule system outside of the testicle, where they are fully matured. They go and finish the process of spermiogenesis, where the acrosome cap forms, the tail forms, the midpiece forms, and then they are ready for fertilization. You should know the acrosome cap. It contains those digestive enzymes to penetrate the egg cell. The tail is there, the flagellum is there to help it be motile. Remember, sperm are not motile until they are outside of the body, until they've been ejaculated, then they become motile. Up until that point, it's just smooth muscle contractions moving them along. And then the seminal fluid that is released with sperm. They nourish the sperm. They have an alkaline pH to help neutralize the acidic pH in the vagina. And it's produced from the secretions of 3 glands: your seminal vesicles, your prostate gland, and your bulbourethral gland, also your Cowper's gland. Remember, you don't have to be super specific with them. I want you to know that the prostate and the bulbourethral glands are secreting mucin for lubrication, and the seminal vesicles have the alkaline fluid, the fructose for energy, and prostaglandins to help with contractions, to move sperm through the male and female reproductive systems. Okay, so that is pretty much chapter 28. Again, I think it seems like I went through it kind of quickly, but once you get into the details of those processes and you get into the hormones that are regulating it, how GnRH, FSH, LH, inhibin, estrogen, progesterone, oxytocin, prolactin, once you get into all those pregnancy hormones and development hormones, you start to realize there is kind of a bit there. So, break it down into the different phases that we're going through: the spermatogenesis, androgenesis. Break it up into the ovarian cycle, the menstrual cycle. Kind of take it in chunks and pieces, and it should be okay. So, chapter 29 for development. Talk about the prenatal period from fertilization to birth. There are 3 phases into that: the pre-embryonic, embryonic, and fetal periods. You should know just in general what is happening in there. During what period are you going to have the most organ growth? Which one is going to be the process of maturation, and the general dates that they're going to be occurring, the weeks that they're going to be occurring. Remember, we said that for the menstrual cycle, for the female monthly cycles, those tend to fluctuate up and down, especially as you go premenopausal and hormone levels start to fluctuate. Those, I don't really worry about the days, but for development of the fetus, that does tend to follow a very strict kind of pattern. So you should be familiar with the general weeks that those are occurring. So the prenatal period, just where development of the embryo is occurring. Usually fertilization, there is capacitation that has to occur. So I look at fertilization as 4 steps. First, it is capacitation of the sperm, which occurs in the vagina and in the female tract, not in the male tract. That's when the nucleus will start to elongate. It loses some of that cytoplasm, and some of the covering on the acrosome is dissolved so that it can more easily penetrate the oocyte. So it has to go through capacitation first, and then it can go through the corona radiata penetration, the zona pellucida penetration. Within that zona pellucida, so it's the corona radiata is on the outside, then the zona pellucida is more is slightly deeper. Once the sperm penetrates the zona pellucida, that's when it has that acrosome reaction. The acrosome cap hits the zona pellucida, and the zona hardens so that no other sperm can fertilize the egg. Because if you have multiple sperm fertilizing one egg, you end up with an excess in genetic material, and it's fatal. And then after the zona pellucida penetration, you have the fusion of the egg and sperm nuclei. Remember, the egg has to finish up its last meiotic divisions, so it becomes an ovum. Then the ovum and the sperm can join. They fuse their nuclear membranes, and they finally have a diploid cell again. After fertilization, you have cleavage. The cell then divides until it becomes a blastocyst. Remember, there is no increase in size at this point. It's just increasing the number of cells. So within the membrane, you have all of these cells that are dividing and developing. Those are going to start to separate into 2 different types of cells, coming both your trophoblast and your embryoblast. the embryoblast will develop into the actual embryo, whereas the trophoblast will develop into sustaining cells. After cleavage, it starts to keep moving down the uterine tube until it gets into the uterus, where implantation will occur. It takes 2 days for implantation to occur, like 3, I guess, days 7 to 9. It starts on day 7, it's finished by day 9. And as that is happening, the trophoblast cells are starting to differentiate even more. They become the cytotrophoblast, which stay more inner to the developing fetus, and the syncytiotrophoblast cells, which are going to fuse with the functional layers of the endometrium. That starts to secrete your HCG, your human chorionic gonadotropin, which is your pregnancy hormone that is detected in pregnancy tests, and is also going to keep stimulating the corpus luteum so that it continues to produce progesterone and estrogen. Should all be familiar. So those are the trophoblast itself is eventually going to become the placenta, but there are other membranes. There's also the yolk sac, which contains all of the blood and the vessels for the developing fetus to nurture it, and the amnion, which is going to provide fluids so that the fetus doesn't dry out. It stays kind of nice and moist. It gives it some more protection. And then the chorion from the trophoblast cells. So the trophoblast cells become the chorion, which eventually develops into the placenta after about 3 months. The placenta has four basic functions: O2 CO2 exchange, waste removal, transmitting immunoglobulins, and estrogen progesterone production. So that is pretty much the review for chapter 29. Again, it's pretty quick because we covered that. It was the last thing that we covered. I spent most of the review time on urinary system just because that was, I think, more confusing and a little bit further away from us. But definitely spend time with all these, do the worksheets. And if you have questions, definitely email me. Again, I'm sorry that this did not work out how I wanted it to. A couple of different mishaps that happened, so I'm sorry, but I will try to be available all over the weekend until Monday, and Monday throughout the day if you need me. Thanks.